All of these pieces of software are free to download, and we highly recommend that you do so. In order to follow this particular tutorial, you will need to download at least the BLAST program and ideally Mega as well. We want to give you the ability to identify your organism from the whole genome sequences, which is usually possible via the standalone BLAST program. If we have time at the end, we want to help you recreate a phylogenetic tree using your 16S sequences that you've acquired from your data. Our sequence providers carry out Illumina 250 base pair paired end sequencing. So they use the Illumina platform. That means that we carry out sequencing from both ends of a DNA fragment. The DNA is extracted and they shear or fragment the DNA to roughly the correct lengths. Each fragment is then sequenced from both ends, creating 2 reads. The idea of this is it's creating twice the number of reads per run and thus more accurate sequencing. The first step really in the process, once the reads have been generated, is that they are trimmed, which removes any adapters. For each sample, you will find 4 files, and they are numbered 1 and 2, which are the paired reads, and U1 and U2. So for all the U1 reads, they are missing their complementary second read, and vice versa for U2. So the 1 and the 2 have both reads from both ends within the file, and U1 and U2 only have 1 read, and they are missing the second. When you submit Illumina reads to NCBI, for example, you can submit the paired reads, so 1 and 2, but you can also submit unpaired libraries as well. So these are FASTQ files, they're raw reads. So what the sequence providers have done very kindly for us is that they've carried out a series of bioinformatic steps, a workflow that takes those reads and using de novo assembly can recapitulate the genome based on those raw reads. So they use a program called SPADES, and that uses the sequence data that you have within each read, and it overlaps them and recreates a contiguous set of sequences. So each of these contiguous sequences that are generated by overlapping reads, they are assembled to create a contig. You will often see the term contig referred to, and that is a contiguous run of bases. For quality control, the reads are then aligned to a reference sequence. And you can see that the closest available reference genome was identified via Kraken, and the reads were mapped to that reference to assess the quality of the data. We've also provided summary data that tells you the sequence depth, the total length of sequencing, for example, and a number of other statistics that might be useful to you. In addition to assembling the genome is also partly annotated, and that's part of the assembly process as well. You're provided with these contiguous sequences or contigs, and you can find those within a FASTA file that is usually labeled contigs or assemblies. So you've got your raw reads, and then you've got your contigs or assemblies. And within those contigs assemblies, you'll find the FASTA file, which is the DNA sequence of each contig, or as called here, nodes. And nodes within this file, they're arranged in size order. So the largest contiguous sequence is the first one that you will see within this file, and the smallest is the last. That's quite important, because if you scroll down that FASTA file, you might find a number of sequences there that are very small or that contain ends that aren't particularly useful. And there may be a circumstance where if you upload your data to NCBI, for example, they require a minimum length of a node. And so you may have to delete those smaller nodes and then resubmit your file. I've mentioned that there are a number of annotation files. Some are perhaps more useful than others. Some of the important ones I've highlighted in red. One that we will be using in a moment is the FNA file, which is a nucleotide genome FASTA sequence. So this contains your nodes, and that is particularly useful when it comes to identifying the 16S or any other specific gene from your genomic data. A very important task, especially if you're new to genomic data and you want a good starting point, we highly recommend that you download BLAST. So this is a standalone program. You're probably very familiar with using BLAST as an online tool. And of course, that's very useful, but it can be extremely useful to be able to generate your own bespoke databases that you can then search against with sequences that you have in hand. And we'll assume that most of you are using Windows computers. The terminology and some of the processes are slightly different for Mac. We also recommend downloading Mega, as I said, for you to be able to recreate phylogenetic trees, which is typically the next large process that you would go through once you've isolated gene sequences. You need to take notice of where this program is downloading to, because you may need the pathway for future operations. We want you to be able to take your own genomic data and to retrieve the 16S sequence from it, and then compare it to the literature and recreate as accurate a phylogenetic tree as you can in order to be able to identify your organism. First of all, we retrieve our 16S sequence from our assembly. In order to do that, we need a 16S sequence of our expected species from GenBank. I assume that most of you either have a 16S sequence in hand that you can use, or you'll be going on to NCBI and downloading the closest 16S sequence. We will then create a BLAST database from our assembly contigs using our FNA data file. So we take that FNA file, the nucleotide sequence file, and convert it into a BLAST database that we can then search for our 16S sequence. Once we've identified which node our 16S sequence is within, we can isolate that node and use it to BLAST against NCBI. So it might sound odd to those of you that are not sort of familiar with this process. We go on to NCBI to obtain the 16S of the species that we think we have in hand. And ultimately, we will end up with the sequence that we've generated through whole genome sequencing, and we will then use that sequence to search NCBI again to refine our taxonomy. The FNA file looks like this. You'll have a series of nodes. So they are a contiguous string of sequence data. They are typically in size order, as I said. So which node contains our 16S? Now, what you will often find is that the 16S will span an entire node. So one of your nodes will likely be the entire 16S sequence. Sometimes the 16S sequence is split over several nodes. So just to sort of visualize the workflow, do you know your 16S sequence of your species? If you don't have that sequence, you can search NCBI for a 16S sequence that is similar to your own organism and download the FASTA file. You can then use those sequences to BLAST against the assembly that we've supplied. Searching for your 16S on NCBI just type in your organism, the organism that you expect to have in hand, which in this case is Bacillus subtilis. You can see here that one of the first hits will be Bacillus subtilis, 16S ribosomal sequence. You can download the FASTA file from there. So create a folder is a good first step whenever you're undergoing an informatic workflow pipeline. Create a folder, save it in a sensible place that you can access easily and you know the pathway to. In this case, we've named our folder 16S test data. And within this folder, we've placed the FNA file and the 16S FASTA sequence that we've just downloaded from NCBI. Now, you will be interacting with this folder via the command prompt. So that's the idea. So being familiar with the pathway of this folder, where it's saved, and the names of the folders and files within it is important. We've now got our 16S sequence, and we want to now use the sequence to blast against the assembly. We need to create a blast database using our assembly. And we use the command prompt for this. Windows computer, just type in command prompt and you'll see the app appear. We want to state the drive. So it'll be dependent on where you prefer to save your files. For us, it's U drive, so we press U and then colon and enter. And in order to open that file, you use \CD and then the folder name and press enter, and that will bring up the specific folder that you've named. And you can see the contents of that folder by using dir or ls if you're on a Mac. We've asked for the contents of that folder to be shown, and here you can see we've confirmed that the FNA file is there. The 16S FASTA file is also there. What we're going to do now is we've downloaded our BLAST program using the link that we've provided. And what you want to do is see the contents of that BLAST program. You can use dir again to view contents, and you'll see all of the tools within the standalone BLAST program. A number of them might be familiar to you, for example, BLAST-N, BLAST-P, make BLAST DB, make BLAST database, for example. You can see there's a number of tools in here that you can call to analyze your data. We want to create a BLAST database, so we're going to ask the BLAST program to create that database of your FNA file. We're going to use make BLAST db.exe as an instruction to the program to make the BLAST database. So in this case, we want to create a BLAST database using the FNA file. So we'll state -in, then the name of the FNA file. And then we have to state what type of database that we're creating. In this case, it's a nucleotide database. So your instruction looks like that on the command. The output will show the numbers of sequences that have been added to the database. So here it says added sequences from FASTA, added 39 sequences. So it appears that it's actually created the database. And you can confirm this by using dir to see the contents of the folder. And now you'll see that within your 16S test data folder, you've now got a number of these database files. Now we want to search that database with our 16S sequence. So in order to do that, again, we can call the BLAST program and we can use BLAST-N. BLAST-N is now going to search against your database. Follow these instructions. So database and the FNA file name. A query, so what are you going to search against that database? And then you state the 16S file name. Remember that whenever you're stating file names, you have to be 100% accurate. So ensure that you've typed it in correctly. And then you need to name it something. So the out instruction is to name that file something sensible that you can then find afterwards. So we've named it the organism 16S blast N, so the operation, and then the file name of the FNA file, .out.txt. And you can see here that that new file, once you've run that, you press enter and then dir, and it's created that output. So then which node contains your 16S sequence? Well, you can open that file. That's a text document within Notepad. Notepad is one of our favorite tools to open text files. It will give you an e-value of the significant hits that your 16S found within your database. And you can see here that node 17 comes out on top. You can see that the identities, the matches are 1,457 out of 1,459. So a very good match. There are no gaps. So you can state with confidence that node 7 contains the 16S sequence. Now you need to identify what it is. You have your sequence in hand. Again, you can open your FNA file in Notepad. You're looking for node 17. You can find it using a Control F, for example, or a search function within Notepad, and highlight that node 17 sequence. Then go to NCBI BLAST and simply paste it into the search box. You can also be more specific to reduce the search space within NCBI and improve perhaps the accuracy and also reduce the time it takes to run the blast. You can select for rRNA or stroke ITS databases to be more specific. And down here, you can state 16S sequences. So you've blast end your node and it comes up with the best hit. As you can see here, we've identified a Bacillus strain that wasn't necessarily the subtilis that we expected, but it has the highest identity and the best match to the query sequence. So we may well decide that we've got a slightly different strain in hand. Very briefly, I want to run through reconstructing a phylogenetic tree using MEGA. Understand where your organism sits in the broader context of the genus, for example. So in this case, what we've done is we've highlighted the top several sequences. You can be selective over this process. That's really up to you. We've just selected a random number here. It's actually the top 29. You can download those by going to descriptions and then selecting faster aligned sequences. And when you download that file, it will give you a text file with all of those 16S sequences. We can see immediately that the titles of these will create a problem when we generate a tree. So at this stage, it's worth taking a bit of time changing these titles. Now, you don't really want to do that by hand, so you can use various search and replace functions. We've listed a number of functions. I won't go into them in great detail here, but essentially you can use these functions to quickly remove spaces and grammar that will cause problems for you later on. Essentially, you want to end up with something like this, where you just have the species and the strain, and you can do that through a number of special search replace functions. Critically, you want to add your node, your 16S sequence, into this file as well. Now, I don't know how many of you use MEGA on a regular basis. It is a very, very nice free tool to regenerate phylogenetic trees. So we would definitely recommend that you explore this program. You can open a file, a session, and we've named our align sequence, including our own 16S align. It's a faster file, so you want to open it in MEGA. You want to press align, and you are presented with this. So you can see here in total, we've got 30 sequences with our node 17 align them using muscle, which is a powerful tool to correctly align sequences. We would generally recommend default settings most of the time and press OK. And now it's found a number of gaps here. It's a proper alignment of those sequences. Save that back in your folder. You can save it as an MAS file. We've now got an aligned set of sequences. We now want to analyze it, and we want to construct a maximum likelihood tree. Again, most of these default settings will be fine for most purposes. We recommend using a bootstrap method. You can choose the degree of bootstrapping that you apply to your data. Typically, you might want to choose 500 to 1,000 times. So it's going to run through the process of recreating the tree, and it will find the highest likelihood. So press OK, and you create a phylogenetic tree that looks something like this. If we have a close look, you can see here that we've got node 17 situated up here, and we might not have appreciated where our organism sits in the context of other strains. So previously we thought that our organism might be sitting in this Bacillus subtilis clade, but actually it's somewhat separated from that. And you can also obviously change the sort of topography of the tree and create circular trees, etc., depending on how you want to represent your data. You can see that we've been very reliant on NCBI, on the existing literature and bank of 16S, specifically 16S sequences. And it is only from researchers like yourselves that these resources exist, and we wouldn't be able to do any of this work without having those resources available to us to recreate these trees and to align various sequences together. So it's really imperative that sequences are shared with the community, and they're shared. It ensures that you gain credit, you maintain ownership of your data, that you can be cited by other articles as well. And they're reported in an accurate way with the methods, etc., that would enable others in principle to recreate your data. And obviously, you're all, I think, very familiar with these concepts.